Professor Nitam V.S. from Professor Dr. N.D. Patil Mahavidyalaya, Malkapur, Perit. I'm here to explain you about the topic structure of chromosome with a reference to the morphology and organization of a nucleosome. Here we go. Chromosome. Chroma means color and soma means body. It is a thread-like self-replicating structure contains DNA within the nucleus of the cell. Chromosome seen during metaphase in mitosis. E. Starberger discovered chromosome in 1875. Later on, in 1888, Welder coined the term chromosome for the first time. The somatic chromosome number is found in somatic cell and it is represented by 2n, that is diploid. Gametic chromosome number is half of the somatic chromosome and represented by n, that is haploid. Chromosome types. There are two types of chromosome. First is autosome. It controls characters other than the sex characters or carry genes for somatic characters, that is skin color, hair color, etc. Next is sex chromosome, which we also known as gonosomes. Chromosome involved in sex determination. Humans and other mammals have six chromosomes, X and Y, also called as heterosome. Females have a 2X chromosome, male have X and Y chromosome. But in birds, the condition is different. The female is heterochromatic, that is ZW, and male is homochromatic, that is ZZ. Here are the numbers with the species of chromosome in a diploid condition. Chromosome number. Diploid and haploid chromosome number. We can differentiate the chromosome by diploid condition and haploid condition. Diploid cell where 2n is the number of chromosome is called is diploid, and in haploid condition, which is n and having only one copy of each chromosome. In animals, gametes, sperm and eggs are haploid. Number of chromosome varies from species to species. Size of chromosome is normally to be short as 0.25 in fungi and birds or long as 30 μm in some plants like Trillium. Structure of a chromosome. Now we will discuss about the chromosome morphology, that is the structure. Metaphase is the most suitable stage to study the chromosome morphology. Chromosome changes its morphology during cell divisions. They are coiled, elastin, and thread-like structure during interphase. When it enters into the mitosis, the chromosomes are highly condensed and distribute to the daughter cell. This is the diagrammatic structure of a chromosome. Each metaphase chromosome appears longitudinally, divided into two identical parts each, and it called as a chromatid. Both the chromosome or chromatid make up a chromosome are produced through replication of single chromatid during synthesis phase of interphase, so they called as sister chromatid. In contrast, chromatid of homologous chromosome known as non-sister chromatid. Centromere, which is also known as primary constriction. Centromere is a landmark of identification of every chromosome. Each chromosome has a constriction point called centromere, which is synonymous, also called as a kinetochore, and it divides chromosome into two arms or section. Centromere position. Size of chromosome. The size of chromosome varies from stage to stage of cell division, as we know. The chromosome has two arms, that is longest and the shortest arm. The longer arm we called as a Q arm and the shorter arm we called as a P arm. The P arm we named as a petitite, meaning small, and the Q arm name is Q just because it follows the letter P in the alphabetical order. Chromosomes are classified according to the centromere position. According to the centromere position, we differentiated chromosome in various forms. That the first one is metacentric chromosome. In this, the centromere is located at the exactly at the center of the chromosome and divided both the arms equally, and it we give a shape of V during anaphase. Next one, that is a submetacentric chromosome. In this, the centromere is located just above the center and it gives one shorter and one a longer arm, and it forms a J or L shaped during anaphase. Acrocentric chromosome. The centromere located close at one end of the chromosome. The centromere is terminally placed and forms very unequal arms. The acrocentric, it is a Greek word acro, means pick. P arm is short and it is hard to observe, but it is still present, and it forms a rod shape during anaphase. Telocentric chromosome. In this, centromere is located at the one end of the chromosome, which is at the terminal end, and that terminality it is called as a telocentric, and it may be also rod shaped during anaphase. DNA and histone together form a chromatin. We will discuss about chromatin. Chromatin is formed by the combination of protein and DNA complex. The DNA double helix in the cell nucleus impacts special proteins that we term as a histone. Chromosome packaging. Now you will think that a larger DNA, how it is packed in a thinnest nucleus. Okay, that is the short region of DNA double helix. It is 2 nm in size. Next one, that is a beads on a string term of chromatin. It's bind in 11 nm. Chromatin fiber of packed nucleosome, that is of size 30 nm. Section of chromosome in a extended form, that is 300 nm. Condensed section of chromosome, that is 700 nm. An entire mitotic chromosome, that is 1400 nm. Histone. Histone can be grouped into 5 major classes, that is H1 or H5, H2A, H2B, H3 and H4. These are organized into 2 superclasses as follows, that is core histone and linker histone. Core histone H2A, H2B, H3 and H4. Linker histone, that is H1 and H5. Linker DNA is double-stranded DNA in between two nucleosome cores, that is association with histone. H1 holds the cores together. It is that if we in a house and we lock the door, that is the lock is H1 linker DNA which protects the house. Nucleosome. It is a segment of DNA wound around histone protein core, repeating of units of eukaryotic chromatin. It consists of 147 base pair of DNA wrapped in 1.67 left-handed superhelical turns around histone octamer consisting of 2 copies. Nucleosome defined as the coparticle plus one of the linker region. Linker histone is H1 and its isoform. Non-condensed nucleosome without the linker histone resemble beads on a string of DNA under electron microscope. Protein DNA structure of chromatin stabilized attachment to the non-histone protein called as nuclear matrix. Heterochromatin. It is a tightly packed chromosome intensely stained and having genetically inactive satellite sequence. Centromere and telomere are heterochromatic. Euchromatin. It is a lightly packed form of chromatin, DNA, RNA and protein. often under active transcription. Unlike heterochromatin, is found in both the cell with nucleus, that is eukaryotic, and without nucleus, that is prokaryotic, active portion of the genome within the cell nucleus. Thank you.